Okay, so we previously talked about amino acids, which are the monomers that give us proteins. Now we're going to talk about the long chains of amino acids and the shapes they get and the different things they can do and how they end up going from a whole sequence of amino acids to a very long chain. Now proteins are extremely diverse, which means they can do a whole bunch of different things. They're biological catalysts as enzymes. They provide the framework for bones. Bones we always think of as being mostly calcium, but it's actually laid down kind of on a protein framework, a protein mesh as it were. Hemoglobin, transporting molecules, moving things across cell membranes, cells communicating with each other, that's proteins. The antibodies in your immune system that prevent you from getting sick, kind of the reason why you never get the same cold twice or you go get vaccines, those are protein. Movement, muscles, actin and myosin, kind of looks like a set of rows in a rowboat moving past each other. That's how you move. Those are proteins. And then also storage. There are some plants that use proteins to store their molecules and store and as a sort of a food source for their seeds, if you will. But basically, what proteins do in terms of what they're involved in basically goes for pretty much everything except for energy storage. Now, they can be used for energy. It's just not primarily what living systems would rather use them for. They're sort of the last ditch result of having to go through all the other systems in terms of energy storage. So if you remember our video on amino acids, they're the monomers. There's 20 different here to make them up. Just like in English we have a 26-letter alphabet. With proteins we're going to have a 20-letter alphabet to make up all our proteins. And just like with words in the English language, some of them are very long, some of them are very short, some of them use a whole lot of letters, some of them don't use very much. You can use more than all 20, so basically you've got an infinite number because you've got an infinite combination in terms of length and numbers and types of the amino acids to put together. The polymer we call a polypeptide. Now we don't call it a protein until it gets a three-dimensional shape. Once it gets a 3D shape and it can function, then we call it a protein. Proteins are the best example of form follows function because they give the 3D shape. We'll take a look at proteins a lot when we do enzymes, but for most proteins, like pepsin here, you can see it's got this notch. That's an active site. It allows it to be a catalyst. We can talk about hemoglobin like you did in grade 11 with its 4 heme rings, allowing it to carry oxygen with those iron cores. That's where it gets its name. You can talk about keratin and collagen, which provide ligaments, cartilage and hair, lots of different structures, but it all comes back to their order of amino acids in those molecules. And so the first level of protein structure is what we call primary structure, but it's the order of amino acids. This is determined by DNA, that you're going to have these amino acids in this sequence, and that's going to give you a chain, what we call a polypeptide chain. Now, if everything else that comes after this is determined by this order of amino acids. So if the primary structure changes, the secondary structure and so on and so forth certainly might change. It's going to make a difference depending on what the amino acid change is. The example that you might be familiar with from grade 11 is sickle cell anemia, where we just change one amino acid, in this case glutamic acid for valine. One of them's hydrophilic, one of them's hydrophobic. It changes the complete shape of one of the hemoglobin molecules. Instead of getting those red toroid donut-like blood vessels, under lower oxygen conditions you get those sickle cell shapes. And in grade 11 you probably talked a little bit about the symptoms and the consequences of sickle cell anemia, and that it allows you to be immune to the malarial parasite. And it's an advantage in certain situations, but it's caused by just simply one amino acid being different out of about 150. Okay. Secondary structure, we talk about local folding. It's an interaction between the amino and carboxyl groups on amino acids. It's usually driven by hydrogen bonding, and depending on whether the hydrogen bonds form every other amino acid or every third, you're going to see two general structures, sort of a slinky shape, which we call an alpha helix here, or sort of a folded sort of paper situation, which we call a beta pleated sheet. And those are two very, very common forms of secondary structures. Now there are other forms of secondary structures besides the alpha helix and the beta pleated sheet. There can be some just electrostatic attraction. There can be some hydrophobic things going on, and sometimes some of these proteins will attract metals to some of their R groups, and that can go on cause some issues too. But overall, alpha helixes and beta pleated sheets are the ones we need to know. A tertiary structure, we call this getting kinky. Basically, this is hydrophobic interactions. There's more hydrogen bonding. There's ionic bonds. We've talked before with functional groups about the sulfhydryl groups, the SH, and how they like to bond together to form what we call a disulfide bridge. So if you can imagine, as you take these alpha helixes and these beta pleated sheets, it brings more R groups and amino groups and carboxyl groups into contact and near one another, and the hydrophobic amino acids are trying to get away from the hydrophilic things, and the hydrophilic things are trying to point towards the outside. The hydrophobic ones are getting towards the inside. The positive ones are trying to get close to the negative ones, and the opposites are going to attract, and all this sort of stuff is going to twist and turn and fold and kink and bend this polypeptide chain into a three-dimensional shape. And then sometimes with some proteins, you need to have more than one strand or more than one polypeptide there, and as they fit together, you're going to get the final functional protein that can do its job. And the best examples we have of that are collagen, where it's very much like a rope and like a braid, where they have to fit together and weave past each other, or hemoglobin, where you've got actual four rings that actually have to come together to form a functional molecule. Okay, so to review the levels of protein structure, the first one, primary amino acid sequence determined by DNA. Secondary, R groups, hydrogen bonding, alpha helixes, beta pleated sheets. Tertiary structure, hydrophobic interactions, ionic interactions in terms of positive and negative R groups, disulfide bridges, and then multiple polypeptide chains with quaternary structure. Now, since proteins are basically built from the interactions of secondary and primary structure, the order of amino acids, anything that disrupts those ionic bonds, those hydrogen bonds, and those disulfide bridges is going to change the shape of a protein, because it's going to change the way it folds and kinks up. And that'll give us what we talk about: temperature, pH, and salinity, or saltiness, if you will. It can also be, so there can be some other things involved here, but these are really the big three that happen a lot. And we talk about when we unfold or alter the folding of a protein, we call it denaturing. And we're changing the shape of the protein, we're altering its 3D shape, and that destroys what it does. The best example that we might be familiar with is the frying of an egg. The egg white is very full of protein, and normally room temperature, it's clear, it's very viscous. You heat it on a frying pan, for example, it gets whitish and somewhat rubbery, and you've actually applied heat and changed the shape of the protein. This is something you can do very simply. You can use pH. You can put lemon juice, for example, on some cut apples. They do this at McDonald's, and it prevents them from browning. We can, put high salt situations, pickle things, for example, and it prevents the enzymes from breaking down or continuing reactions. It's a very common, useful thing, and we'll see the effects of these factors later on, but this is really one of the reasons why homeostasis has to happen. The Goldilocks zone, you have to keep temperature within a certain range, you have to keep pH in a certain range, and salinity, because as soon as you go outside of that Goldilocks zone, you change the shape of the protein, you destroy the functionality, because form follows function. There it is again, kids. And as soon as you change the form, you change the function. So that's the basics of protein.